Good morning, students. We are meeting for the very first time, and as a formality, I would like to introduce myself to you. I am Dr. V.S. Reddy. I am heading the Department of Botany. Along with me, 3 staff members are working on permanent basis, and other 3 staff members are working as a management paid candidates. Totally 7 staff members are there in our department. Out of 7, we are 4 engaging in the classes. First of all, at the outset, I am going to welcome you all to the Department of Botany. It is one of the oldest departments in the college. The department was established in the year 1965. Since then, it is imparting quality education to the students, and there are very good alumni belong to this department. And many students, they made a very fine career from this college. Even I hope you will also be one among them, and you are going to make a very fine career. About the department, I am going to introduce in brief. First of all, we have the 2 laboratories for the BSc students, and we have one research laboratory which is going to help the students to make or conduct a very small projects that we are going to give to the students in their regular curriculum. Even we have the nursery attached to the department. In the nursery only we are going to procure the plants which are needed for the day-to-day practicals. Most of the plants that we need for the practicals are usually brought from the nursery itself. We have a well-maintained herbarium. About 750 plant specimens belongs to about 100 different families have been preserved over there. And some of the plant specimens belongs to endemic category, endangered category, as well as some of them belongs to critically endangered categories. We have also got the museum. In that museum, the plant specimens belongs to diversified fields of botany have been preserved. We have well-maintained medicinal plant garden, and in that medicinal plant garden we have cultivated many plant species of medicinal importance. Even we also provide very small projects to the students, and we are assisting the students to expose themselves to the research. Even we help the students to inculcate their research ideas after going to their MSc or even their studies. Definitely this is going to be helpful for the students. And we got the grants from various funding agencies. We got grants from UGC, then VGST. Even we also got the grants under other programs. Even Department of Forestry help us to build a very good nursery. You will come to know all of these facilities when you really come up to the campus. As far as the BSc first semester is concerned, really the topics mentioned for the BSc first semester are really interesting, and I hope you will be in semester program now. So far in PUC, you are exposed to annual examination scheme. but it's not like that. In semester scheme, there will be 4 months teaching, and 1 month is there for the examination, and 1 month is there for the holidays and other activities. Within 4 months program, you are going to write 2 internal theory examinations and 1 internal practical examination. These are all in addition to the semester and examinations conducted by Karnataka National Vidyalaya. That's why always at every 15 days or a month you will have an examination. And whatever the marks that you are going to score in these exams will be pumped to the university, and the same will come in your mark sheet. That's why you are not supposed to neglect your studies. Always you have to be alert, and don't think that after 1 week or after 10 days I am going to make the study. Nothing like that. If you have the habit of postponing the things, definitely it is going to harm you. And you may not be able to make up. That's why you have to be very regular. You have to be very regular in your studies. And all the staff members of our department are very coordinated with the students. Whenever you come across with any doubts, any clarifications you want, definitely we are going to help you in the academic matters. As far as the BSc first semester is concerned, the BSc first semester, we will study about the topics such as microbiology, then algae, fungi, bryophytes, pteridophytes, and gymnosperms. These are the 6 units. Microbiology is the first unit. Algae is the second unit. Fungi is the third unit. Bryophytes will be the fourth one. Pteridophytes the 5th unit. And the 6th unit is the gymnosperms. The final theory examination conducted by the university will be there for 8 marks. And theory internals. Internal marks will be some based on the performance that you are going to do in the 2 theory internals. Even the assignments that we give, your attendance will also going to give the prominence here. Totally for theory internals, 20 marks are. Altogether, botany theory examination for the semester 1 is there for 100 marks. As far as practical subjects, practicals will be conducted on these topics that you are going to study there in the theory. And practical examination will be conducted for 50 marks. Out of 50, 40 marks will be there for the semester end exam. Okay, and 10 marks will be there for the internals. Altogether, the botany subject for the semester 1 is going to carry you 150 marks. As far as my topics are concerned, I am going to take up some of the aspects belongs to the microbiology, and the entire unit of the fungi I am going to consider. These are the two topics that I am going to deal, and rest of the topics are going to be taken by other staff members. Professor Gadi will take some of the classes, and Dr. Srimati Ratnayasan will also take some of the classes, and another staff member, Vijayanand, will also take some of the classes. I am going to deal with the very first topic. That is the donated for me. That is microbiology. Microbiology. You know about the biology. Biology is the branch of science. It deals with the study of living things. Micro refers to small. Micro refers to small. Microbiology, it's nothing but branch of biology deals with the study of microorganisms. Microorganisms means what? Microorganisms means there are very small units. Very small living things. Very small living things means very small living organisms. Smaller to the extent that we could not see them with naked eye. We can't see them with the naked eye. To that extent, they are small. Usually, we are able to observe the things which are having the size of about 0.10 to 0.90 millimeter. You can see the size of the organisms belong to this particular range. There are living things or organisms which are smaller than this much size. When the things are smaller than this size, we are not able to locate them with our naked eye. We are not able to observe them with our naked eye. Such organisms which we could not observe them with our naked eye are considered to be very small. And these smaller living things or smaller organisms are usually called as microorganisms. One asks the question is that if we are not able to observe the microorganisms with our naked eye, how we could see them? How we could observe them? We are going to see them with the help of an instrument. That instrument is usually referred as microscope. It is a device or an instrument. It has the capacity to magnify the object to several hundred times. Maybe it can magnify the smaller things to the extent of 100 times, 200 times, even 1,000 times, even sometimes to the extent of 1,00,000 times, even more than that. Based on the magnifying power, we are going to identify the microscopes. Simple microscope, compound microscope, electron microscope, like that. Different microscopes have got different magnifying powers. By observing these organisms under a microscope, we are able to recognize their size, their shape. Even we can also study their metabolism. These microscopes help us to study about the microorganisms. Okay, there are different microscopes that we are going to use in our day-to-day studies. One of the microscopes is saline microscope. Second most commonly used microscope is compound microscope. Then the electron microscope. Phase contrast microscope. Fluorescent microscopes. Like that. There are different microscopes. In our laboratories, we are having these two microscopes, light microscope as well as the compound microscope. With the help of these light microscopes, you can see the microorganisms. You can see the smallest things like cells, parts of the cells like cell wall. You can see. Even sometimes cell organelles like plastids also you can see with the help of these simple and compound microscopes. Electron microscope is the one which is usually used extensively for the research purposes. Not only these microscopes are costly, at the same time it's quite difficult to maintain. That's why only research institutions where enormous research work is going on is going to be equipped with electron microscopes. Then let me consider what are the different types of microorganisms. What are the different types of microorganisms? Microorganisms are not all same. There are different types of microorganisms. The very first kind of microorganisms are usually referred as the algae. Second type of microorganisms referred as fungi. Third type of microorganisms may be the bacteria. And the fourth type of microorganisms are usually referred as the protozoans. Okay, these are different types of microorganisms. Other than this, we also consider viruses. Also as microorganisms. But truly speaking, viruses are not organisms. They are treated as altogether different compared to these microorganisms. Later I will explain why viruses are treated as a separate category things compared to all these microorganisms. They are not fitting to the microorganisms. Because viruses have got their own characteristics. With the help of those characteristics, they are different from rest of the microorganisms. But because of their smaller size, even viruses are also going to be studied under microbiology. Okay, these are the different kinds of microorganisms that we are going to study. To study about the algae, there is a branch of microbiology. We usually refer that branch as microbes. Like that, to study about the fungi, again there is a branch of microbiology, we call it as microscopy. To study about the bacteria, there is a branch called bacteriology. And to study about the protozoans, there is a branch called animal microbiology. And to study about the viruses, there is a branch called virology. In addition to this, there is one more branch there in microbiology. We refer it as parasitology. It is widely studied under microbiology. Parasitology. Okay, it deals with the microorganisms lives as parasites. There are some microorganisms which live as parasites. And such organisms are specifically studied in a branch that is called parasitology. This is with reference to that of what are the different kinds of microorganisms and where do we study these microorganisms and what are the different branches of microbiology. Some basic information that I would like to give you about the microbes. I am not going to deal with all the microbes, remember, and along with me of members will also teach about bacteria, fungi, even the viruses. Okay, you will not only study all this, but I am going to deal with the viruses. The term virus, it is actually derived from Latin language. In the Latin language, virus means poison. You all know about the poison. It is some toxic substance. Whenever it is there inside the body, definitely it is going to harm the body. Even certain poisons are lethal to the extent that they can kill the organisms. Poison is something that is going to cause harm to the organisms. Like that, whenever viruses are there inside the body of the organisms, definitely they are going to harm. Certain viruses are going to cause mild diseases. Certain viruses are going to cause some severe diseases. Diseases are going to be severe to the extent that even organisms are going to be killed. That's why we refer virus as a harmful thing, or it is just similar to the substance that is poison. First of all, what we say is viruses are ultra-microscopic. We say them as ultra-microscopic. Ultra is very small. Very, very small. Ultra-microscopic means they are very small things. Very, very small things. Even they are smaller to the extent that they are several times smaller than bacteria. They are several times smaller than bacteria. Here I am going to give you one simple hint. Size of the bacteria is usually measured in terms of micrometer. Remember, 1,000 micrometer is equal to 1 millimeter. Okay. Viruses are said to be smaller things. Smaller to the extent that yes, we can measure them very conveniently with the scale, that is micrometers. Whereas viruses are concerned, they are still smaller. And size of the viruses can be usually measured with a nanometer scale. We measure them with nanometers. We say them 10 nanometer, 20 nanometer, 100 nanometers, like that. And remember, 1,000 nanometers is equal to 1 micrometer. I'm just giving you the scale. Then you can imagine They are literally very, very small compared to the bacteria. And I am going to give you one more simple comparison. You can see the bacteria under compound microscope. Bacteria can be seen with the help of compound microscope. You can observe the bacteria, you can see the bacteria, even you can measure the size of the bacteria with the help of compound microscope. Suppose if you want to observe the viruses with the same compound microscope, you can't see them. You can't locate the viruses with the compound microscope. The reason is they are very small. Even they are invisible under compound microscope. If at all we want to see the viruses, then we are going to make use of electron microscope. They are visible only under electron microscope, because you all know that electron microscope is the device which is having very large magnifying power. Because of the electron microscope ability to magnify the image of the object to several lakh times, yes, we can see the size of the viruses. Not only viruses are said to be ultramicroscopic, even viruses are said to be the particles of the organic matter. They are said to be the particles of organic matter. Particles of the organic matter means what? Particles of the organic matter means the viruses are made up of organic compounds. If you collect the viruses, analyze them chemically, you will come to know that yes, the body of the viruses is made up of organic compounds. Okay, what are the organic compounds which are there in the body of the viruses? Body of the viruses is usually made up of what you call as the nucleic acid and the proteins. Usually there in the viruses, one can see either DNA or RNA. Both the nucleic acids are not present. If there is a DNA present, then RNA is absent. Then if RNA is present, then DNA is present. Out of the two nucleic acids, any one of the nucleic acid is present. It may be either DNA or the RNA. Okay, assume that this is supposed to be the nucleic acid. Maybe it is a DNA. Okay. The DNA is going to be there in the body of the virus. Around this DNA, that is nucleic acid, there are proteins present. Look at this. These may be the proteins. These may be the proteins. They are supposed to be the proteins. I am just considering the generalized structure of the virus. For that matter, you take any virus, yes, basically it contains a nucleic acid in the center. It is going to be surrounded with the proteins. This is supposed to be the coat or the cover made from the proteins. You will find nucleic acid and proteins. That's why viruses are said to be the nucleoproteins, because they are basically made up of nucleic acid and proteins. These are organic compounds. Viruses are constructed with the organic compounds. That is why we refer them as particles of organic matter. Remember, they are not called as an organism. Viruses are said to be particles. Why they are called as particles? Because viruses do not possess cellular structure. There is no cellular structure in viruses. No cellular structure. There is one norm. If at all anything is to be called as an organism, that thing should be made up of what you call as cells. Cells should be there. What is meant by cell? Cell is nothing but a very simple compartment-like structure. Within this compartment-like structure, yes, there is a protoplasm present. Yes, this is usually the protoplasm, and this protoplasm is usually covered with a membrane-like structure. We refer it as plasma membrane. Suppose if it is a plant cell, then surrounding the plasma membrane there is an additional coating present in the form of a what we regard this as cell wall. Suppose if it is an animal cell, then cell wall is absent. What you will find is you will find only the plasma membrane as an outer covering present surrounding the protoplasm. And in this protoplasm, all the metabolic activities which are needed to keep the cell alive are taking place. Maybe the cellular respiration, maybe the protein synthesis, maybe the lipid synthesis, maybe the synthesis of ribosomal amino acids. Yes, all the activities which supports the life are going to occur there in the protoplasm. And inside the protoplasm, yes, we may see the nucleus, even we may see the other cellular elements like mitochondria. Even sometimes we may see what you call as Golgi bodies, endoplasmic reticulum, lysosomes, ribosomes, everything. If you look at the virus, in the virus, no protoplasm of its own present, and there is no outer covering present in the form of plasma. There is no cellular structure present. If a cellular structure is absent, then we are not going to treat that as organism. If you are not going to call it as an organism, then what you can call it? You can call it as an entity or a particle made up of organic compounds. Entity made up of organic compounds can also be called as a particle of organic compounds. And another important thing that you have to remember about the viruses are said to be always multiplying. They are multiplying in the host cells. They multiply inside the host cells. Suppose assume it is a plant cell. It is a plant cell. By chance, if any plant virus, if it infects the plant cell, then always there inside the plant cell, this virus is not going to keep quiet. once it enters in the host cell, it is going to multiply. It is going to multiply and it is going to keep on reproducing. By the way of reproduction, it is going to enhance its number. One virus becomes 2, 2 becomes 4, 4 becomes 8. Like this, this number simply increases. It's possible only when the virus is there inside the host cell. Suppose if the same virus is there outside the host cell, what happens? If it is there in the air, if it is there in the soil, if it is there in the water, if it is not in contact with any living host cell at that time, it behaves as a inert particle, inactive particle. It never multiplies, it never grows. But if the same virus comes in contact with the host cell, proper host cell, immediately it infects, it enters into the host cell and it starts multiplying. And remember, viruses are not able to live for a very long time in absence of the host cell. If at all they want to multiply, if they want to spread, they want to go on pandemic scale, they need the proper host cell. Only in presence of a proper host cell or proper host organism, they multiply. That is why viruses are usually referred as obligate parasites. We call them as obligate parasites. Compulsorily for their survival, for their multiplication, host cell is required. There is a host cell and only they use inside the host cell as a parasite. They derive all the benefits from the host cell for their multiplication. If the host cell is not available, then in absence of the host cell, they behave as a nut. Okay, based on this, one can frame the definition of the viruses. How one can define the viruses. Let me consider the definition of the viruses. Definition of the viruses can be given like this. Viruses are ultra microscopic